So lab exam 2. It's going to be via Proctorio. So on Thursday, we're going to finish up the last of our material with the reproductive system, and we'll take a break. Then we'll go into the Proctorio for lab exam 2. And then the next time I'll see you will be Tuesday for exam 4 in person at College of Marin. So this
is the lab, it's open book again though?
open lab notebook.
Okay, so it'll probably see us looking down a lot and reading.
Exactly.
Okay.
Open lab notebook, but not like random notes. You know, and your lab notebook should not be filled with random notes either. But, and again, you will be turning in your lab notebook on Tuesday for the final grading of that as well. So this is going to start, with the rods and cones lab. So the basic concepts from before are still important. You should know what independent and dependent variables are, what qualitative and quantitative are, what correlation is, and things like that. But in terms of the actual practical labs that you need to be familiar with, we're going to be starting with the rods and cones lab. In grading this one, I was surprised at how many people kind of missed one of the main points in their kind of discussion and analysis of the data on this one. So I think it's worth talking about this one for a few moments. So what was our independent variable and dependent variable for the rods and cones lab? You remember we had this stick, you're dropping the stick under different light conditions and seeing how fast you can grab the stick.
The independent was the light intensity and the dependent was the speed of your visual reflex.
Mm-hmm. So exactly. So this was kind of the reaction time of visual reflex. So we varied the light and we saw how fast you could grab the stick. And what did we find?
That the light density did, or the light intensity did impact the reaction time.
And in which way?
The more light, the faster the reflex.
Yeah, we saw that the brighter the light, the faster the reflex, the shorter the reaction time. And that made sense because we talked about cones being processed faster and cones being used in brighter light and rods being slower and the rods mediating dim vision. But there was sources of variability. What was an important source of variability we brought up that we wanted to make sure we controlled for, or at least checked into?
Making sure that that you didn't get better at it as the lab went on.
Yeah, exactly. Practice in general. The more you do some task, usually the better you get at it. Just practice. Practice makes you better. And as we keep grabbing the stick, it could be that is an important variable that is actually affecting reaction time that's not light. So how did we design the experiment so we were able to dissociate those two things, like be able to look at light versus reaction time versus, as opposed to like just the amount of practice you've been getting, versus reaction time?
We randomized the light intensities.
And we randomized the light intensities. So then we could analyze the data from two different viewpoints. We could do brightness, from like dim to bright. And then we had reaction time. And we saw a really nice negative correlation. The brighter the light, the shorter the reaction time. But because we randomized the trial numbers versus the brightness, we could also look at, okay, what was reaction time and trial number. And we saw it went up and down and up and down. That it was not correlated. It was pretty random, which makes us feel comfortable saying, okay, this trial number, the amount of times you've been doing it is not such an important thing determining the reaction time, especially compared, looking at brightness, we saw this really nice, clean relationship. Dimmer, especially when you average the different subjects across the class, you got this nice dimmer the light, longer reaction time, get brighter, just kept getting faster. Whereas trial number, it jumps up and down. Some people got a little, I think, confused. They said, well, it looks like it's on average going up. That's not really the point here. The point was it's not correlated. It's jumping all over the place. So I think that's something I really want to make sure everybody really takes away from this lab, was this dissociation of this confounding variable or possible confounding variable. We designed the experiment so we could analyze the data and look at practice. And it's nah, that doesn't look like that's important. Brightness, it's woo, yeah, we got a really nice relationship. Right, so that was an important part of this lab, was kind of seeing how, if possible, try to design the experiment so you can look at the data from different ways. And if we had just made, each one was brighter and we had something that looked like this, we wouldn't know, is this due to practice or due to brightness? But because we randomized the brightness with respect to trial, we can analyze the data and say, brightness correlated, trial number, which would be practice, not really correlated. So does that make sense? So that was the first one here. What else did we look at? Hold on, let me go into my, oh no, it's slow again. What was the next? Oh, then we did all the blood pressure stuff and the blood stuff. Blood pressure, you should know how to take blood pressure. We learned about how you take blood pressure and why that technique works, that auscultatory method for the indirect assessment of blood pressure. Remember, you had somebody's arm and you put a cuff on it. When you took a stethoscope and you're listening to stuff on their brachial artery. When you first pump up the cuff, you don't hear anything here at the brachial artery. Why is that?
Because it's fully compressed, there's no blood moving.
Exactly. If you pump up higher than the highest pressure, higher than the systolic, there's no blood flow, so you don't hear anything. There's nothing flowing. And then as you lower the pressure, what happens as soon as the pressure in the cuff gets below the systolic pressure?
You start to hear sounds because some of the blood is moving through, but it's still sort of compressed, partially compressed.
Exactly. That's where we get those sounds of Korotkoff. So as soon as you're below systolic, blood is able to kind of squish through, but it's compressed. So it's kind of going. So you can hear that turbulent flow. And as you keep letting the pressure get lower and lower, at some point the sound disappears again. What is going on when all of a sudden the sounds of Korotkoff stop?
And diastolic volume, right? Or and diastolic like pressure, right? Then you can, that's the last beat, right?
And so that's how we know we are now below diastolic pressure. And why does the sound disappear?
It's freely flowing.
Yes, that's smooth laminar flow, which doesn't make any sound. Like really smooth flow is silent. So make sure you understand how we measure blood pressure and why it works. What's the significance of those sounds you hear, right? And then the other thing was we looked at a couple of things. We looked at exercise. What did we notice that happened to your blood pressure and pulse in response to exercise? Yeah, they went up, which makes sense. You got to increase your cardiac output, increase the flow to supply all the extra needs in response to your muscles, and actually in this case, going into oxygen debt recovery. Did blood pressure, like systolic, diastolic, increase kind of similarly?
No,
Now, what did we see?
the diastolic didn't change as much or much at all.
The diastolic didn't really change much at all. It was the systolic was the one that went up. So make sure you're like looking at your data and kind of remembering that. Whereas in, what else? We looked at pain. What's the effect of pain on blood pressure and pulse?
Increases it, it's gonna make it faster. And your blood pressure higher, right? Because you're basically in like panic mode.
Right. So again, what does your intuition say? What does our data say?
This was the ice part, right?
Yeah, this is the ice, the cold pressure test.
Everyone sort of had a different reaction.
Yeah, different people had different reactions, right? The part of the reason why I really like this one is because most people have that intuition of like, oh, you're panicking, you're gonna go, if you look at the actual data, a lot of the heart rates went down. Blood pressure, we had the hyporeactors that didn't change much at all with the blood pressure. Hyperreactors where it changed pretty dramatically. That's an important point to take home from this lab, is that there's a lot of idiosyncratic responses to pain. And some people react with huge changes. Some people react with very small changes.
I have a question about that. We did that part of it on the heels of the exercise test, and so some of the heart rates going down, I would intuitively think that they're just going down because the person just...
We used different subjects, though. They weren't the
that's
same
right.
subjects that were doing the exercise.
So the subjects from the earlier class did not just get off the Stairmaster.
Correct. There's like 4 people in a group, and one person volunteers to do the exercise. Another person volunteers to do the ice water bath.
Gotcha.
Yeah, no, otherwise, if you hadn't waited long enough, that could be a problem. What about nicotine? What does nicotine do? We looked at results if people are smoking.
It increases heart rate and blood pressure, including diastolic.
It increased. And we talked a bit about what's kind of the mechanism of increasing the blood pressure.
Vasoconstrictor.
Yeah, we talked, it's a vasoconstrictor, which means you're gonna have higher blood pressure, increased resistance. Vasoconstriction means higher resistance, and blood pressure is cardiac output times resistance. So if we're increasing heart rate and increasing resistance through vasoconstriction, we're gonna increase our blood pressure. So at least acutely, we're gonna increase blood pressure due to these mechanisms. In the longer term, it's other stuff going on. There's inflammatory responses causing, heart disease and things like that. But even just in the moment of smoking, you can, kind of increase stress on your cardiovascular system by just vasoconstriction, increasing resistance to flow. Okay, we have 2 minutes here. Blood type. And we did the blood lab with, you should know how to figure out blood typing. Like if I did a blood, let's say it looked like this. What kind of blood type am I?
B.
I am type B because the anti-B antibody serum is agglutinating, which means it's recognized the B antigen. So I am type B. Who can I give blood to?
AB
Yeah, AB. AB is the universal recipient.
and B.
B, other B people. Who can I receive blood from?
O and then B.
Yeah, I can get B as blood and O, the universal donor. You should make sure you understand how all that works. So it's 12:30 right now. So I realize it's 12:30. I'm going to keep doing this review for the people who can stick around. If you have to go for whatever reason, this is going to be recorded. So it will be available on, in Canvas and before the evening is done. Also, if you have questions about things as you keep studying, I can respond to emails, but I do acknowledge it's 12:30, but I am going to continue on with the review right now. So feel free to stay with me here. The other thing with the blood lab, we looked at hematocrit. Now, how did we measure your hematocrit?
Through the centrifuge.
We put the blood in a little tube and then we put it in the centrifuge and we spun it down. And at the end of all the spinning, there was going to be some plasma left at the top. The cells all get pulled to the bottom and we can just measure that ratio. And that was the hematocrit. What did we have to worry about? What kind of tubes did we use to do hematocrit determination?
Heparinized.
Yeah, we had to make sure we used the heparinized ones because otherwise, as we saw, the blood coagulates, the fibrin forms, and it's all going to turn into jello. You're not going to be able to spin the blood cells down. As opposed to the non-heparinized tubes that we used when we wanted to check out how long it took for blood to clot. When we put the blood in the non-heparinized tube, after a few minutes, you break the tube and you've got some weird stringy stuff starting to form in there. What is that weird stringy stuff?
Like the platelet plug? No.
Fibrin.
Fibrin.
That's fibrin. That was the fibrin that was forming, stuff that makes the clot. Did we see anything that affected how long it took for the blood to clot?
Aspirin?
Yeah, so what was the effect of aspirin on blood clotting?
It took way longer.
to clock.
Yeah, if you take a look at your data, it was somewhere around like double. So yeah, aspirin, and this also makes a difference. Like if you get, wheeled into the ER, and they're like saying, are you on any drugs or this or that? Having somebody who's about to cut you open or deal with you all kind of like, cut up, it makes a difference if they know you're on aspirin or not, because you're going to bleed out a lot easier if you're on aspirin versus not. Hemoglobin, we looked just with that hemoglobin with the little filter paper, that Tallquist test. Okay, we then, what was the next lab we did?
Respiratory.
We did the respiratory labs. So we did exploring the respiratory system. When you breathe out, how is the air different than the air you breathed in?
It's warm, moist.
Say it again.
I said it's warmer and it's moister.
It's warmer. And what are the implications of that in terms of, just basic physiology as we've been talking about it?
Insensible water loss.
Yeah, exactly. That was another thing a number of people did not address in their discussions on this lab. I really wanted you to kind of just connect that every breath that you breathe out, you're losing water, and that is part of your insensible water loss. That's why maintaining water balance requires that you're taking water in. And even if you are desperately trying to conserve every last drop, unless you live on Arrakis and own a nice still suit, you are losing a lot of water to the environment just by breathing. So exploring the respiratory system, we looked at the holding your breath. If I wanted to hold my breath the longest, what would I do ahead of time?
Hyperventilate.
I'd hyperventilate. And why is that going to help me?
You blow out more carbon dioxide.
Exactly. So that's another thing for holding your breath or CO2. People, again, in their discussions sometimes talk about taking in lots of oxygen. It's like irrelevant, absolutely irrelevant. What is going to determine whether or not you feel the need to breathe is how much CO2 is building up. In fact, if you overdo it, you can hold your breath so long that the oxygen runs out and you pass out before you feel like you need to breathe. So it's not about oxygen, it's about CO2. So that was important. Right, yeah, no, looks like Amazon is down again because I can't get into my stuff. Dagnabbit. All right, what other labs did we do? Oh, spirometry. So spirometry, one of the things that people, most people got, but some people didn't, so I want to review briefly. What do we call just the volume of if this is volume in liters, and this is just time, as time goes by, this breath volume as you're breathing. What do we call the volume of just a normal, quiet breathing in and out?
The tidal wave,
Almost.
tidal something.
Tidal volume.
Volume.
Tidal volume.
Let's wave.
So our tidal volume, or actually it's like a V sub T. Again, usually around a half a liter. And then what is inspiratory reserve volume? Where would I measure that from where to where?
That's that you would do the beginning of the normal inhalation to the one where you breathe in as much as you can. So that high peak.
Yeah, so it would be this. This difference from a normal inhale to as much you can inhale on top of that.
So
some
from the
people
end
might-
of a normal inhalation, right?
Correct. Some people kind of messed this up and they kind of went from here. They said it was this distance, which is not. It's from the top of a normal inhale until the most you can breathe in. The ERV is kind of similar, but for the exhalation. It's the most you can, a normal exhale down to the most you can breathe out. But make sure you understand where you measure these volumes from. And also remember, they are always positive. It's always a positive number, the absolute value. So if you're going to calculate somebody's vital capacity, which is just the sum of all three of those, it's just adding three positive numbers. It's adding their tidal volume, which is a positive number, their inspiratory reserve, which is a positive number, their expiratory reserve, which is a positive number. Some people got into trouble by making the expiratory reserve a negative number, which then gave us a wonky answer for vital capacity. We looked at the forced vital capacity where you then we had the machine kind of measure for us how much of that did we get out in 1 second compared to the whole. So this whole thing here would be FVC. FEV1 is the amount that you're able to blow out in the first second that you're blowing it out. And how are these relevant for trying to assess somebody's respiratory health?
If the ratio is too low, then it means that they're having trouble getting air out of their lungs. So it's obstructive. It can be like a marker for obstructive pulmonary conditions.
Right, exactly. So we looked at that ratio, the FEV1 over FVC. Typically it should be, greater than 70 or 80% or so in order to, for normal respiratory function. If it's less, then that's indicative of some kind of chronic obstructive pulmonary disease, like asthma or something that's restricting the flow and making it harder to get the air in and out. So you should understand this ratio and why we care about it and what it is, how to measure it. And then the last thing we did in that lab, cough. What's distinctive? If you were now that you've done this lab, if you're going to describe a cough to somebody in a more like sophisticated way, what could you tell them?
The amount that you're coughing out doesn't change from a normal exhale, but the speed at which it comes out does.
Exactly. So we saw that, if we look at the flow, when the cough happens, there's a big negative flow, meaning very fast liters per second leaving. But if you look at the volume, you're breathing in and out and in and out. It's like it goes out fast, but the volume that leaves during the cough is not really different than a normal quiet exhale. So it's very fast, but the volume is not different than a normal exhale. And why, what's the practical ramification of this? Why not just try to really get all your air out and try to blow this piece of steak out of your throat?
In case you need to cough more after, you need to save some.
Yeah, exactly. We are not using it all. We're kind of using a fast impulsive blast to try to dislodge something. And if that didn't work, we got quite a bit of air left over. We can do it again and again. Right? Eventually you're gonna run out, in which case you're gonna like turn blue and die unless somebody saves you. But yeah, cough. Yeah, I think it's fascinating. I mean, the first time I did this, I kind of intellectually knew, and then you look, it's like, oh yeah, no, that's real. That's kind of, again, for me, non-intuitive. That's one of the things that's really good about empirical data. You know, looking at the actual universe rather than just assuming it must do what I think it does, right? You know, when you look at the actual world and do the experiment, sometimes you realize, oh, maybe I was wrong in my intuition, my gut sense. Sometimes your gut sense is good, sometimes your gut sense is not good. So, what else did we look at? Trying to think about these labs. What's that?
Urinalysis lab.
Yeah, the urinalysis. You know, multi-sticks. You should be able to look at a multi-sticks and read it and interpret it. So I'll just say that. Right, those are the little things, like, you know, each color symbolize something, and depending on the color, it would mean different things. What are things that you can measure with the multi-sticks?
Glucose.
Glucose, what other kinds of things? How much glucose should you actually see in your urine?
Pretty much nothing.
Pretty much nothing. It should all be getting reabsorbed in the proximal convoluted tubule. If you do have glucose, what is it likely a sign of?
Diabetes.
And diabetes, like if something's wrong with insulin, right? We just talked about insulin. If either type 1, you're not making the insulin, or type 2, your cells are not responding to the insulin. In either case, glucose is building up in your bloodstream, not getting absorbed by the cells. And if it gets up to high enough levels in your bloodstream, the kidney tubules don't have the capacity to reabsorb it all before it makes it to the far end of the tubule and it comes out and shows up in your urine. We also saw things that were, if you had a urinary tract infection, what kind of stuff might you find?
The leukocytes.
Very likely to see leukocytes. What other things might show up?
Blood.
Blood might show up. What other kinds of things?
The nitrile?
There's nitrites that were metabolic byproducts of bacteria that are often responsible for UTIs. Yeah, so you should know multi-sticks. The kidney lab. Let me, hold on a sec. I can't go onto the, website or our Canvas because Canvas is down, but I can go into my files. And oh man, I don't, hold on.
I have it downloaded. I can share it in the
Oh, can you do that? That'd be great. Thank you. I'm going to stop sharing to allow you to do that.
Do you want me to share my screen or do you want me to
No,
put
just share your screen.
hold on, sorry, let me open it. It's loading. I have a Mac, so it might look a little bit different.
Okay, and can you take us to the page with the graphs there? Right, so take a look here. No, so this is perfect, thank you. So which group is that brown line at the top there?
Or 3.
That's group 3. Why does it make sense that group 3 had the highest sodium chloride concentration?
Because they ate all the salt and they didn't pee much out. So whatever there was full of salt.
And why, just theoretically, why does that make sense?
Because they had less to dissolve the salt.
Wait, I'm sorry, say it again.
They had less water to dilute the urine and dissolve the salt.
So there was less water. So just because there's less water means you're going to have a higher concentration. Whatever's left in there is going to be high concentration. Why was there so little water?
Because they only drank 80 mils as compared to the 800.
Right. And I guess the thing I keep wanting people to get,
Osmosis.
Salt was holding on to the water.
not osmosis. So, and osmosis is going to be part of the mechanism, but in the bigger picture, just
They're
thinking
trying to
about,
maintain osmolarity.
yeah, it's trying to maintain your water balance and your blood osmolarity. So if you are not drinking much at all, particularly if you're eating salt, your body is going to conserve water and not pee it out. So we're going to expect very small urine volumes. You know, again, what the takeaway message from this lab I really want you to have is this idea of, you know, your kidneys are working at water balance and at salt balance, basically preserving the osmolarity of your body, right? So if we need to either hold on to salt or let go of salt, or hold on to water or let go of water as appropriate to keep our homeostatic osmotic level. So it makes sense if you're not taking in much water at all, you're going to have to conserve that water, so not much pee. All 3 other groups, actually, Bonnie, can you go to the volume one? You know, you can see all 3 groups that ate or drank 800 mils of water all had pretty huge volumes of urine output, because you're diluting yourself to maintain osmolarity. You got to let go of that water, as opposed to group 3 where it's conserving water. You know, there's slight difference there. We saw group 2 that took in the water and the salt is not letting go of quite as much water, which kind of makes sense, because if you got extra salt, you're going to have to hold on to a little more water to keep your osmotic balance, right? Here we saw that caffeine kind of sort of increases, you know, output beyond what would be typical, although it's subtle. You know, caffeine is a slight diuretic, but it's not a dramatic diuretic. If you want a real diuretic, you would take like a loop diuretic, something that shuts off the sodium pumps in the ascending limb of the loop of Henle and destroys the gradient in the kidney medulla. Then you're going to pee a lot. How about that one, NaCl amount? This one's also kind of fun. This is basically showing the amount of salt your body is getting rid of. You know, group 2 got rid of more salt than group 1, which makes sense because group 1 didn't eat any salt. Group 2 did eat salt. So to maintain your homeostasis, you're going to get rid of salt if you take it in. But the weird thing on this one is that the caffeine group is also getting rid of salt. So in fact, I think that's part of the mechanism, I think, of caffeine being a slight diuretic is by increasing salt excretion, and the water follows the salt. Again, group 3, even though you ate the salt tablets, didn't have a huge amount of salt excreted because you just didn't have a lot of urine to carry the salt out of your body. In order to get stuff out of your body, you need urine volume. So specific gravity, again, is going to be pretty related to just the salt amount of the salt concentration is going to be mirroring the specific gravity. How did we measure specific gravity for this lab as opposed to the multi-sticks?
There's the float.
Use that little float, that urinometer. So when we put that little float in there, if there was high specific gravity, like we would see on that brown line, the float would be floating up very high out of the water or out of the urine, because the urine is more dense. If the urine is more dilute, low specific gravity, the float will be lower. So that's another really common way to measure specific gravity. The last thing we did was the digestion lab. There were 3 parts. There were carbs, lipids, proteins. For each of these, you should understand the assays we used to measure things. Like again, it's a lab practical, so there are pictures of things, just like any of these things we've been talking about that actually have physical things. There might be pictures that you have to interpret. Pictures could be graphs, but pictures could be test tubes. If I have a test tube in my lipid assay that with my litmus cream and it looked like this, what could you tell me about the condition of the fats in there?
They haven't been broken.
They have not broken down. The whole point here, if the fats were breaking down, what would I see?
It'd be pink.
Exactly. Because you can start getting the fatty acids forming, and the fatty acids are going to make the pH go down, which is going to make the litmus powder turn pink. So we have this assay by looking at how pink or blue. What kind of conditions gave us the best fat breakdown?
Bile salts, and I forget what the other thing is called.
I can tell you what it is. The pancreatin, which was the enzyme, the lipase. Right. So the lipase can break the fats down, but what was the role of the bile salts?
Kind of breaks them into smaller pieces.
Yes, or to hold them into smaller pieces, to emulsify. So as you kind of shake up fats, like the bile salts, I wouldn't say break it into smaller pieces, but kind of helps it stay in smaller pieces. Right. So as a fat breaks into little droplets because you're shaking it up or stirring it, the bile salts can get in between, and the fatty part or the non-polar part will be near the fat, the polar part will be near the water, and now these little droplets don't have the urge to all regroup into one big glop again. So what conditions were best for protein breakdown that most mimicked like the stomach?
Acidic.
Acidic, and what else did we have?
Pepsin.
Yeah, we needed to have the enzyme as well, but then the enzyme works best if it's in an acidic environment that mimics the stomach, right? It's one of the Normally, low pH is going to denature an enzyme, but in the case of pepsin, what denatures pepsin really?
Lack
Yeah.
of ability.
So it's not going to be as happy in neutral, and it's going to be really unhappy under what kind of conditions did we see?
Alkaline.
Alkaline, like the sodium hydroxide. How did we tell if protein was breaking down?
The egg white got smaller.
Yeah, the little egg white, the albumin gets smaller, kind of just breaking apart and dissolving. And for carbs, where did we find an enzyme that could break apart carbohydrates? Right?
Your mouth.
Saliva,
Saliva, the salivary amylase.
right? And what evidence did we have that salivary amylase really does break down carbohydrates? Sure, I can tell you that, but what was the evidence you saw, at least from we could have doctored the videos, but we didn't.
The Lugol's and the Benedict's.
More specifically. So you have
Altos, yeah. And then
Or starch.
this, yeah, both starch was present.
wait, so I heard lots of little, so somebody kind of say the narrative more clearly.
Lugol's was purple if starch was present, but yellow if there was no starch present. So the breakdown would be, it would be yellow,
And so
showing.
where did we actually see evidence, actual evidence that amylase does indeed break down starch? Which tube?
In
Which
the
tube?
presence of maltose or lack of.
Which tube was the one that really convinced you that saliva actually genuinely has the ability to break down starch? You have your lab note.
Oh, the one that was just pure, one that was just pure starch, and one that was pure maltose. Is that what you're talking about?
Yes, like the starch and like what showed the most breakdown was the starch and saliva.
Right. So I'm asking what it, so yeah, I think Bonnie's on the right track. So I'm asking, I want evidence. So sure, I can say amylase breaks down starch, but what is the actual evidence that that's true that you saw?
In the tube that only had the starch and saliva.
And what happened in there?
It was,
The solution turned brown.
or yeah,
What do you mean by that?
The Benedict's turned brown for presence of maltose.
Brown. I'm not sure it was brown. I think it was more of an orangey, but
I get those two tests confused.
but you definitely want to know those tests. I mean, those tests could show up, right? You need to know if you see test tubes that have had Lugol's or Benedict's, you need to know how to interpret what those mean and, figure out what happened in that tube. So when we looked at Lugol's, if we took saliva and put it together with starch and let it just incubate for a while, and we tested it with Lugol's, what did we find?
It was
Turned yellow.
And which means what?
No starch is present, so it had been.
Exactly. So the starch disappeared. And then if we tested with Benedict's, what did we find?
It turned that reddish, dirty yellow positive for maltose.
Right. So we saw, so that's what I'm talking about. There is evidence that saliva is doing this, what I'm saying. It's not just me yapping at you. It's like you can see like, okay, oh wow, mix starch with saliva and the starch disappears and the sugars appear. So there we have more actual evidence of amylase doing this rather than just having to trust me. And like I said, in this world of doctored videos, I could have made that up, but I didn't. You could do
Is
it.
this
on-
lab one of the ones you're going to be going over in person? Because I didn't see an assignment to upload the.
Yeah, exactly. I'm gonna just grade this one in person. What what can put the brakes on amylase?
Acid.
Acid.
Like environment.
Yeah, so what did we see? What happened? What happened if we changed the pH radically and made it really low pH? What happened to amylase and its ability to work?
Denatures.
Denature. And so what did we see in terms of the kind of starch and maltose at the end of a half hour incubating?
In glucose, it was still blue, meaning that there was still starch present.
Yes,
And
the
it
starch-
Benedict also.
Purple.
so the starch did not break down and the maltose never showed up. So again, we have this evidence that, oh, amylase did not work under those conditions. Whereas we saw very different results if we let amylase work on the starch in just, more neutral conditions. So are there any other kind of questions or things to think about or review?
I know that the one of the cardiovascular labs, we kind of ran out of time, but for the heart sounds and pulse in class, I think that was like towards the tail end. All you want is kind of like what we did on our partner for the heart sound.
Right, and some of those, the things you could have done on yourself at home, right? Feel for your own carotid or your own things. So,
But all you're looking for when we turn these into eventually is just like I wrote down my observations of when I did them. And like what
yeah, and yeah, which ones could you feel well, which ones could you not feel well? Right, yeah. What else? I am looking here if there's anything else I missed.
I just have a question about time for the lab test. I know last time I felt like I was running a little short and that's kind of stressed me out. If we feel like we're running out of time, is there a way to get that extended, or are we just like, that's it?
So the reality is I have to be, in general, there is a limit, I would say try to just finish it in the time you've got.
Okay.
But I do my best to give you enough time. So, and that is part of the studying is having a reasonable sense, not having to like learn the stuff on the fly.
Yeah, of course.
Also, if I'm remembering right, we couldn't go back. Once we finished a question, we had to just keep going forward. We couldn't go back to like check our work or skip a question and come back to it. Is there a way that you can change that, or did you do that intentionally for a reason?
I did that intentionally, so people don't like kind of go down and like, again, over the last 2 years, I wish it wasn't true, but there was a lot of cheating and there's a lot of people searching stuff, collaborating with each other. It's been horrible. I'm so excited to just have our main exams back in the classroom. And next semester, I spent so much time just documenting cheating so I could to my dean over the last year of online exams. It's been horrible. So it's just one of the ways to try to minimize that. And it's not saying anything about you, but it's saying in the bigger picture that if we don't try to, do at least these imperfect ways, it just gets even more out of hand.
If we finish our final and there's some time, are we able to because I know we can't see the lab exam once it's done, can we look at it on your computer in person that day or?
Once everybody's turned it in, yes.
So Tuesday when we come in for the final, after we're done with the final, we could look at.
Absolutely, yeah.
Okay.
Yeah, absolutely. On Tuesday, you can you can definitely, we can go over your exam for sure.
And do we then not get back our lab books ever?
You get them back anytime you want to come by and pick them up once we
Okay.
hit the hit,
Great.
hit, yeah, the spring semester. So yeah, no, a lot of people like them back and find them useful in the future. Minimally, they've put so much work in, they're not quite ready to just let them disappear yet.
Exactly.
Our new coffee table book, just anyone who comes over gets to read our lab books.
Cool. So any other questions or anything?
So for the multi-sticks, you had like practice ones on there for us, and there's patient A, B, C, and D. Are the answers somewhere just to
No,
verify?
you can ask me right now, though.
What was patient D?
Let's. You could ask me if the Amazon gateway is back.
Oh,
The Amazon gateway is still. Dagnabbit.
it's still down.
Let me see if I have it in another folder. That's. Yeah, it's down. It's so frustrating. Okay, hold on a second. I'm gonna go into your.
But for that, you would like show, if that's like on the lab test, you would show us like a picture of it. We don't have to be like, you wouldn't just be like, this is this color blue. What does that mean?
No. So Irene, I found. Tell me which patient it is.
You can see.
Wait, no, tell me which one. I found
Patient D.
D is in doorknob. Okay, here it is. Let me share this. Okay, so no glucose, good. No bilirubin, no ketones. Specific gravities, fine. Ooh, what do we got? We got lots of blood. pH is fine. Protein is fine. Urobilinogen's fine, negative. Yeah, so what's wrong with this person?
Period.
There's nothing wrong?
Oh, there's something major wrong.
Oh, they have blood.
Lots of blood. What are the possible, what's your differential diagnosis for patient D?
Menstruation.
So
Menstruation.
Could be
I thought
contamination from
just
menstrual
to,
blood.
could it be contaminated?
Right, so that's right. We're saying it could be contaminations from menstrual blood. If it's if it's a woman of reproductive age, you'd check in with her.
Okay.
Or it could be, what other things can cause blood in the urine?
The UTI.
UTIs can do it. What other kinds of things?
STD.
That I'm not sure.
Menstruation.
That one we've come up with already. Contamination.
Something's wrong with the kidneys.
Right, something wrong. It could be, again, you were like in a bar fight last night and got just a major shot to your lower back, and your kidneys are damaged right now. You can also get it from I had a student once who ended up getting blood in their urine, I think from some kind of a over-exercising thing. So there are other things, I'm not sure. There's definitely other things that can cause blood in the urine, but as a rule, blood in the urine is concerning. If it's just a contamination, then that's not a problem. But if it's actually real, it's definitely something you need to follow up. So blood in the urine is not good. But yeah, so that's the kind of stuff that you would, could be showing you this and saying, what is anything out of the ordinary here, and what might you think is wrong if you saw this? So that could, I wouldn't say could, there will be stuff like that on your lab exam. Who am I fooling? All right, let's see. Any other questions or anything? All right,